Hi all, Frank Lynch here. I regret that I'm unable to join you for our faculty introductions today. I had a conflicting doctor's appointment, but I made this video to introduce myself to you. I'll tell you a little bit about myself and a little bit about my background, and then I'll talk about a couple of the projects I've completed and a couple of the projects that I have that are ongoing. So I grew up not far from Cheney in Ritzville, Washington. I got my undergraduate degree at Linfield University. It's a smaller school in Oregon where I was a double major in mathematics and physics. After Linfield, I left to go to graduate school at the University of Utah in Salt Lake City. It was there I got involved with the mathematical biology group, and I'll talk a little bit more about that in a second. Prior to being employed by Eastern, I was a faculty member at a school in Los Angeles called Occidental College, and I've been here at Eastern since 2013. Really happy my path led me back here. I'm really proud of the work Eastern does and who we are in our community. So I'll talk a little bit about biomathematics or mathematical biology. My definition of biomathematics is an interdisciplinary field that uses quantitative mathematical tools to investigate biological processes. So a typical strategy might be that one has a look at a physical system that they're interested in studying. You might determine some mechanistic questions out of that system, write down some models that represent that system mathematically and analyze them. And then it's important at the end to link your mathematical analysis with some sort of biological conclusion. So you're looping around to explain what the math says in terms of the physical system. So I'll talk first about a project that I was involved in studying hydraulic conductance. So I show this piece of celery not as a system I studied, but rather as an example of xylem flow. So this is an experiment that a lot of you might do in middle school. You put the celery in some water with food coloring, and of course the celery sucks up the food coloring water in its xylem. And that's the system I was looking at. So specifically, I was looking at a plant called Guzmania lingulata. So Guzmania lingulata is a plant that grows in the tropics of Costa Rica, and the faculty member I was collaborating with at Occidental College was interested in understanding how these plants conserved water in their xylem. So it turns out that this conservation effort gives plant physiologists an idea of how plants are able to survive in arid environments. So we were mostly looking at flow in what's called the blade of the plant. So these bromeliads sort of gather water in their tank area, and then that water is sucked up into the tank, into the plant through the tank. It moves in the axial direction, sort of parallel to the growth of the plant into the blade, and then it moves out in the radial direction so it can do photosynthesis with the leaves. So I talked a little bit about that decomposition, but again, what we're interested in here is thinking about hydraulic flow into the blade through the blade tank interface, up through the xylem of the blade, and again the axial direction, and then out to the surface of the leaves in the radial direction in the blade. So as a first try to model this, we modeled the blade part of the plant just as a rectangle between z equals 0 and z equals L, where z equals 0 corresponds to the blade tank interface and z equals L corresponds to the top of the blade, the end of the leaf. So it turns out you can derive pretty quickly sort of an unusual boundary value problem. And that boundary value problem is a second order differential equation in the potential U, where we have values prescribed at z equals 0. There's a no flow condition at z equals L. And we prescribe this additional additional value at 0 that the flow is all in the axial direction. Now, I mentioned the flow that the plant physiologist is interested in. So in their lab, they can actually measure sort of a total flow through the plant. And what we're interested in doing is breaking that flow apart into components in the radial and axial direction. And once again, the component in the axial direction gives you a quantifiable measure of how a plant is able to conserve water. So in this boundary value problem, you can see it's a little bit unusual in the sense that it has a second order equation with 3 boundary conditions. Now, as it turns out, the constant A in the differential equation is unknown, and solving for that constant is equivalent to solving for the flow in the axial direction only. So it's equivalent to solving that decomposition problem. So as it turns out, in this first try, the function g that controls the geometry of the blade is pretty simple because we have a rectangular blade, and you can write down an analytical solution that satisfies the boundary conditions. One can then go ahead and use a numerical method in order to determine the A that satisfies this additional boundary condition. Now, we weren't exactly satisfied with this approach, so we thought a more interesting approach might be a second model of the geometry of the leaf, where instead of assuming that the geometry of the leaf is rectangular, we assume that the geometry of the leaf is triangular. So in this model, instead of a function g that's constant, we have a function g that looks like some sort of first order polynomial in the variable z. So it turns out, and this is pretty cool, that you can write down an analytical solution for this system too, and that analytical solution involves some sort of archaic airy functions, but nevertheless, one can solve analytically for the function u, and then again go ahead and use some sort of numerical scheme, again like a Newton method or something that solves for the constant a that satisfies this additional boundary condition at 0. And again, solving for that constant a is what our colleague plant physiologist wants to know. She wanted to know, the conductance in the axial direction. We were a little dissatisfied by this approach too, because certainly the leaf is as much of a triangle as it is a rectangle. So even more interesting approach was to try to model the edge of the blade with some piecewise cubic splines. So what we did was take a photo of this leaf, incorporate the data points that make up the boundary, and then go ahead and use some cubic splines to have a representation of that geometry. So in this case, the function g that controls the geometry of the situation is no longer a function at all, but it's sort of just some numerical data that we can use to solve or approximate the solution of this differential equation numerically. So we did that too, and we again were able to solve for the constant a in this case that represents the flow in the axial direction. A couple different publications for this, including a publication in the Letters of Biomathematics not too long ago. So I'll talk a little bit about a second project I've been involved in. This is a project that examines competitive binding. So in particular, we're interested in what's called a mutation in mitochondrial DNA. That mtDNA stands for mitochondrial DNA. So as you may know, DNA has all of these T's and A's and C's and G's that are bound together, and it might be the case, as you can see from the illustration below, that there's a T where there should be a C. So when there's a incorrect DNA point at one spot, we call that a single nucleotide polymorphism, or a SNP. So it turns out that that SNPs can be markers of pathology, and it turns out that we're interested in mitochondrial DNA because, at least at the time I started this project, it was more tractable. So mitochondrial DNA has on the order of tens of thousands of base pairs, whereas human DNA has on the order of billions of base pairs. So, identifying SNPs in mitochondrial DNA that are markers of pathology is a more tractable problem. So the model of this is using a microarray. So a microarray is a small chip. It's about a centimeter by a centimeter that has a bunch of spots on it, illustrated by these circles. These spots have little probes of DNA that are designed to match to a wild type DNA, the WT from the previous page. And also, since a SNP only differs in one spot, they'll also bind to the SNP. Now, as it turns out, what happens is that you run a microarray with a soup of SNP and a soup of wild type, and one of those is marked with a fluorescent signal. So the signal that you get out of such an experiment might look like this. And the amount of fluorescence is sort of a marker for how much of the target, or you can think of that as a wild type, the target that is bound. And you can imagine that the more competitor or SNP you have in the soup, the less target would be bound, so the less the fluorescent signal is. So a typical competitive signal as a function of time, as these combined, might look like this, where you can see the blue line is an example of no competitor, and then the more competitor you add, the more that signal decreases. Now we can write down some differential equation models for this type of a system, analyze those differential equations, and when you do that, you end up looking at what's called a nonlinear system of ordinary differential equations that looks like this. Now, in this system of differential equations, the state variable b superscript t stands for the amount of target that's bound, and the state variable b superscript c stands for the amount of competitor that's bound. And of course, at the beginning, there's no target or competitor bound, which gives us some initial conditions. So an interesting question to ask here, and this is sort of the question that we were addressing by writing down this model, is given some sort of fluorescent signal, can we tell how much competitor is in the soup? So that sort of gives you an idea if the competitor is a marker for these SNPs, whether there's a remarkable amount of mutated DNA that could be a marker of disease. So this resulted in a publication too. This has been published in the Cody Journal. There are a couple interesting limits that you can look at in the parameters of the model that give some interesting results, and I'll leave it to you to check that out. Okay, finally, I'll wrap up with a few ongoing projects. So in collaboration with Nick Burgess in biochemistry, I've been working on this metabolic pathway for DITP. It turns out at the end of this metabolic pathway, DITP is cleaved into DIMP and incorporated into the DNA, and just like this polymorphism problem, the amount that gets incorporated in the DNA can be a marker of disease. So Nick and I are working on some ODE models and maybe some parameter estimation that will give us an idea of what's going on with this system. I'm also interested in ion transport problems. So an ion transport problem specifically with regard to a semi-permeable membrane. So as it turns out, when you have a semi-permeable membrane in ion transport, it lets some of the ions through but not others. And although these ions like to be electrically balanced, they fall out of that balance nearby the membrane. So there's sort of an interesting PDE model that requires some perturbation theory and possibly a stretching transformation to have a look at concentrations near this boundary. And I'm pretty optimistic that this model that I'm looking at will lead to some interesting results with regard to sort of a different background charge on one side of the membrane than the other. And finally, I'm interested in the problem of tree trunk growth. So in this cross-section of a tree, there are two main parts of a tree. There's the heartwood that provides structural stability and the sapwood that provides a delivery system for nutrients for the tree. As the tree grows, of course, it gains rings, and those rings in sapwood become heartwood. And I'm interested in sort of a partial differential equation model of this tree growth, which might end up interestingly being what's called a free boundary problem. So unlike a lot of systems in partial differential equations where the spatial domain is well established, here you can imagine that with a growing system, the spatial domain on which you are looking at this model will grow also. Generally speaking, free boundary problems are hard to solve, and in particular, I think there might be also sort of a Stefan problem involved in this model as well. A Stefan problem is a problem where you have sort of two distinct phases, and we have those two phases here in the sapwood and in the heartwood. So other things I'm interested in this tree trunk growth is what happens when a tree is injured, maybe by insects or birds or by a lightning strike, and how it recovers from that, and then how those rings continue to grow in relationship to that injury. So again, I expect there to be a partial differential equations model here that will give us an idea of how to model this tree trunk growth, but there's still some more to unpack here. So that's it for me. I'll go ahead and acknowledge some of my collaborators, including Gretchen North, my plant physiologist from Occidental College, Dr. Nick Burgess, a number of students I've worked with, and those students have received support both from the National Science Foundation and from the Barry Goldwater Scholarship. So thanks for your attention. Again, I apologize to miss you today, but I'd be happy to chat with you anytime. I'm not hard to find in my office in Kingston 311. Have a great day, folks.